got a little ahead of myself last time because I was a little behind on myself. I started lecturing on number 1, eukaryotic transcription, when I should have started lecturing on eukaryotic transcription, part 1. So we'll backtrack a little bit. I gave you the introduction about structure of a gene and how things work. So that's generally what this introductory lecture is about. So we'll go through it fairly quickly, and you'll notice I'm behind on getting rid of the blue and all the shadows and the crazy fonts and stuff so you can have it easy to print out. So that's an ongoing struggle. So here we go, eukaryotic transcription and general principles here. Of course, it occurs on chromatin, not on naked DNA. You have to decondense. It turns out that most regulation, or a great deal of it in a eukaryote, is simply dealing with the condensation state of the genome. It's such a big component of regulation where it's a very minor component in bacteria. Eukaryotic polymerase cannot read, it can read DNA, but it doesn't know where a promoter is. You know, in bacteria, when you have the holoenzyme, which includes one of the several sigmas, that unit called RNA polymerase has enough information or enough structure to read and recognize a promoter. The eukaryotic polymerase is a little larger, but it certainly has more subunits. It has 12 subunits, and still it doesn't know what a promoter looks like. In a eukaryote, you have the identification of where a promoter is conducted by a series of complexes called basal transcription factors, whose job it is to seek out promoters. And there's an ordered sequence of binding. But it could be because the eukaryote promoter is so masked by histones that you need to do it ahead of time. It takes a while to find the promoter. You sort of have to pre-stage the identification of the promoter, opening up the chromatin, getting everything ready for the arrival of RNA polymerase. Basal transcription factors, these are general transcription factors, not specific to any one particular gene. Their job is to assemble at the core promoter, and the core promoter is defined as those DNA elements that cluster close to the start of transcription. Basal transcription factors. Usually it's contained within about 100 base pairs of the first nucleotide of transcription. And so it turns out in a eukaryote it's much easier to identify a promoter simply by location, because it's always right at the beginning of transcription. And it's easy to tell where transcription starts, whereas it's hard to tell where transcription starts in a bacteria, right? First of all, the messages don't last more than a minute and a half, 3 minutes or so. So they're very unstable, keep turning over. And even more importantly, it's the rule rather than the exception to have several coding regions all contained in a single transcript. So you never know which one of those protein coding regions has in front of it the promoter. You know, if you see a series and they're all pointed the same way, it may be at the very beginning, the first one that's beginning to point in the same orientation, or it may not be. It could be a couple, and then it'll have an internal promoter and start again. It's just very hard to predict where the promoter is in a bacterial cell. In eukaryotes, it's pretty easy. You see a gene encoding a protein, the promoter will be at the start of transcription. The core promoter, I say it's contained within about 100 base pairs. That's actually a little larger than what you usually need. you can encode it essentially in about 50 base pairs or so. And then, but you normally have the sites for transactivators to bind, transactivators being distinguished from basal transcription factors. Basal transcription factors nucleate at very close to the start of transcription and form the pre-initiation complex, which eventually includes the RNA polymerase itself. All the other kinds of elements are for binding of transactivators or repressors. And the sites for the binding of those controlling regulators is highly variable. Usually you have room for a couple of binding sites within 100 base pairs of +1 of transcription. So that's the most influential position, certainly in a plant. And then you have these clusters of controlling elements called enhancers that can be located also very close, or they can be located inside the transcribed region, inside of an intron, or they can be past the 3' untranslated region, or they can be 100,000 base pairs away, especially in a metazoan. Plants haven't learned the trick of how to place a controlling cluster of regulatory elements that far away. So here you see the basic diagram that I've been describing here. Basal factors bind the core promoter. It's usually considered to be contained within the first 100 base pairs from nucleotide 1, or the start of transcription. Transactivators may bind in clusters to cis elements, or they can bind individually, not clustered. They are still binding sites for transactivators, but they're not called enhancers unless it's a multi-element cluster. Depending on the composition of the cluster, what kinds of transactivators are clustered together for their binding, you will have different developmental and expression outcomes. This is where the gene expression specificity is obtained. Of course, the enhancer clusters, or the individual elements, can work in either polarity, flip or flop. If you have something forming the pre-initiation complex, though, because they are so intimately tied to direction of transcription and the binding of particular proteins in a particular order, a lot of times these elements in the DNA are fixed with respect to position. Just like in a bacteria, you have fixed positions for -10 and -35. These help establish the polarity of transcription, as they do in bacterial promoters also. Core promoter, then the upstream or distal promoter, together it would be called the full promoter or something. A little more jazzy, I can't think of it at the moment. Okay. Before we get, like last time we went into the details of Paul 2, we need to take a couple of steps back and get a bigger picture before we zoom in and spend our rest of our time on Paul 2. So one thing about eukaryotes is, even though they started in archaea with a single RNA polymerase, that's how it all started. Over time, and apparently very quickly, eukaryotes begin to specialize their RNA polymerases themselves. So now you have a minimum of 3. Plants have 4 or 5, but most things have 3, and they're specialized to different categories of genes. Paul 1 is for the ribosomal structural RNAs, RNA polymerase 1. Paul 2 is for protein encoding genes. And Paul 3 is for a variety of small RNAs, mainly the tRNAs. There's a 5S ribosomal there too, and some other small ones, kind of a grouping of odds and ends for Pol 3. Pol 1 has a specific location or address in the nucleus. It's clustered around the ribosomal genes, as you would expect, and they in turn form this kind of gelatinous concentration called the nucleolus. It's not surrounded by a membrane, but it's something that diffracts light, and you can just look through a light microscope and you can easily see the nucleolus in most cells. Core promoters. How are these three different RNA polymerases, what's the structure of their promoter? Well, in most cases, for polymerase 1 and 2, it's just as you would expect. It's centered on the start of transcription, a little bit upstream, so the polymerase doesn't get in the way of its own transcription. But surprisingly enough, Pol 3, even though it has several different promoter types, its most predominant promoter type is where the elements of the promoter are actually internal to the region transcribed. You can delete all the way up to +1 and even go further, and you will not affect transcriptional activity. That was a big surprise. We'll see how that works in a minute. Of course, there is one class of Pol 3 promoters that looks more traditionally, looks sort of like a Pol 2 promoter, but more about that in a minute. Pol 3, the mRNA is the least abundant. It's 2 to 5%. Oh, it's messenger RNA. Pol 2, the one that we love to talk about in lecture and to study and to speak in hushed, reverent tones. Pol 2 only represents 2 to 5% of the total transcripts. Most of it is being pumped out by the other two polymerases, these more simple-minded polymerases, to talk down to them. They just need to know what 1 or 2 promoters look like, and they're just like phage. T7 doesn't need to know but 1 promoter, the phage T7 promoter. These specialized 1 and 3 have a fairly simple job in finding a promoter because they're not a lot of variation in them. Most active is Pol 1 because it's ribosomal cisterons. Second most active, Pol 2. That's kind of hard to reconcile there, to look at that more carefully. Okay, let's look at the subunit structure. Here you see it says greater than 10. It's actually 12 subunits in typical eukaryote. What you'll see is, though, you'll see the amino acid sequence signatures of those prokaryotic RNA polymerases. You'll see something that is very similar to beta prime and beta, even in the archaeal, even in the eukaryotic RNA polymerases. You have greater than 10 is actually 12 subunits. I was in a laboratory when I was in graduate school at University of Georgia that was trying to isolate all the subunits of RNA polymerases and make antibodies and do fundamental transcription studies in plants, which is a big mistake. It's burned through many careers because it's so hard to work with plants. But anyway, so we lived and breathed RNA polymerase. Here's a feature of Pol 2. We're on Pol 2 now, looking at subunit structure. And the largest subunit, that would be the one corresponding to beta prime, wouldn't it? The largest subunit, as I've mentioned before, has an unusual feature. It has 50 or more, well, depending on the organism, 20 to 50 copies of 7 amino acids over and over and over in an unstructured C-terminal domain. As you may suspect, this is a signaling post. It is modified, phosphorylated primarily, to represent the different stages of transcription. It also serves as the binding site for big complexes that are dragged along behind the RNA polymerase. It's like the polymerase is the pickup truck and it's pulling like 4 boat trailers in a row behind it to take care of what all the fish is going to catch that day, to process its RNA primarily. This is only a property of Pol II, to have this great long carboxy terminal domain of the largest subunit, Pol II. Sometimes I'll give an exam where I will give them a great long amino acid sequence, a 500 kilodalton, not 500, but about 150 kilodalton protein. I'll say, what protein is this? And they go, really? Why didn't you ask me which way's north or something? I could have answered it just as easily. If you look at the back end of it, you'll see over and over and over and over again a repeat of 7 amino acids: serines, prolines, threonines, serines, prolines, threonines. The only thing that has that is RNA polymerase II, largest subunit. Do you find that in archaea? Nope. You don't find it in E. coli. You don't even find it with the other RNA polymerases. And why is it there for Pol II? Because Pol II has special needs, in terms of processing for its transcript. Putting the 5-methyl cap, removing the introns, terminating, even though the polymerase keeps on going, adding poly A. All these things need to be following along very close behind the wagon for Pol II. How about our ancestral RNA polymerase before 1 became 3 or 4 or 5? What did that look like? Well, it didn't have a CTD. We go all the way back to archaea. But how many subunits did it have? Did they look anything like the eukaryotic, complicated 12 subunit? Well, the answer is it was just as complicated, and even more so. It had 13 subunits, and it turns out that it's really the same polymerase. It split the largest subunit, beta prime, into 2 proteins, but otherwise it's the same. It's really 12, the large one is in 2 parts. We inherited this complicated RNA polymerase from archaea. It's a clear signature, you know, people who talk about evolution and things like that, they're always looking for, you know, to drag their knuckles, do they stand up? They're looking at the visual, but if you look at the molecular evidence, you see it is just rock solid all the way back to the Hadrian period. Enough preaching. And here is that repeat. You see the serine, proline, threonines. I've forgotten what Y is. Anybody remember what Y is? Tyrosine. Yeah, tyrosine. No sigma, of course. No sigma. What would take the place of sigma? Well, sigma was involved in promoter recognition, so some of the functions of sigma would be perhaps performed by the transactivators that are binding to enhancers and other far upstream or the distal promoter. But sigma does other things too. It melts the promoter. How does a eukaryotic polymerase melt the promoter? TF2H. There are other things. How about this abortive cycling? Does a eukaryotic Pol II undergo abortive cycling? Yep. Is anything that looks like domain 3.2 of sigma poking its head in the RNA exit pore? Have we finally escaped that moray eel of the nuclear depths? Nope, it's still there, poking its head, being bumped and bumped and bumped until it gets bumped out as a whole nother protein, or not to be, or to be. TF2B does that function. You'll see some similarity in terms of structure, but no amino acid conservation in one of the basal transcription factors, TF2B. And TF2B, did eukaryotes invent that? Nope, Mama Archaea has a TF2B. All this stuff comes from Archaea. The polymerase with its complexity, Tata binding protein, TF2B, TF2E, S, F, it goes on. Okay. One of the polymerases is sensitive to a fungal toxin. You've heard of the, what's it called, the angel of death mushroom, alpha amanita. It's a red mushroom with white flakes on the top, deadly poison. It kills RNA Pol II specifically, and it was used in the early days to characterize the kinds of polymerases that you were trying to purify. You'd look for its sensitivity to alpha amanitin, Pol II. Some people over in the micro building used to order that, and of course we ordered in the lab I was working with. Pol II, sensitive to alpha amanitin. They all can transcribe DNA non-specifically in vivo. So if you just put DNA in a test tube with the polymerase, which is what I naively did as a graduate student, you'll get a ton of transcription. And that's because at every little nick and every little random place, it will just start transcribing. RNA polymerase is the happiest when it's transcribing. So most of regulation, it turns out, is figuring out a way to prevent RNA polymerase from being happy, from transcribing. Paradoxically, it's not coaxing that polymerase to transcribe. No, it's to hold it off and just open the door just occasionally. We got room for one more. It's all in keeping it from transcribing when it shouldn't. Okay, Pol 1, then we'll do, I don't know, Pol 3, I think, and then Pol 2, and we're through with the introduction material, and we can get diving into TF2D, which we started on last time. Pol 1, this is where it lives in the nucleolus. This is what its promoter looks like. Of course, you don't need to memorize this, but you're going to have to know the controlling basal factors that control it. So it has a core promoter located close to the start of transcription, and here you can see it with my pointer here. Notice it only goes out to about -40. Looks a little bit like a bacterial in terms of length. And then it looks like a hard-boiled egg here or something. This is an upstream binding factor, UBF. And well, the binding factor is, UBF is the element, binds the upstream upstream binding factor, SL1. I don't know why it's called UBF and down here called SL. On the exam, you're going to need to know the term SL1. SL1 is the basal transcription factor that binds the, I got it. It's been a while since I've seen the slide. The core promoter is bound by SL1. The upstream promoter element is bound by a second basal transcription factor called upstream binding factor, UBF. UBF controls the magnitude of expression, but it cannot determine where the RNA polymerase should start transcription. That is controlled by the basal factor that binds the core promoter, and that's a theme. You'll have two general categories of basal elements sometimes. One that is responsible for determining the start point for transcription, where nucleotide number 1 should be. That is called the, and I hate to say it, commitment. Commitment. It's the commitment factor. Guys don't have those. Yeah, they do. The commitment factor In each of the promoters, you need to know what is the commitment basal factor. It's the one that determines where the start of transcription should be. And notice our friend, TATA binding protein is a part of the commitment factor for Pol 1, although there's no TATA sequence at all for it to recognize. SL1 has several subunits, one of which is TBP. It also has a more distal element, the upstream promoter element, and the protein or complex that binds there is called the UBF. Core binding factor, TBP, 3 others, function to position the polymerase, directs a low level of constitutive transcription, and of course that is boosted up by the UBF, which binds at the distal element, or the upstream element. It ensures high frequency amplitude. The UBF seems to wrap DNA around it. What does that look like, wrapping DNA around it? Like a nucleosome, doesn't it? Is this a nucleosome that got smart and figured out what promoters look like? We'll see that apparent wrapping around in Pol 2 also. We don't quite have that figured out, but it's an observation people have made for a long time. Name 2 factors that regulate transcription from Pol 1 promoters. 2 factors. Factors being the proteins, that would be number 2, looks like to me. Yep. Number 2. Which of the following is responsible for determining the start point, which is the commitment factor, not the upstream binding factor, but the SL1? Pol 3. Do Pol 3, then we're on to Pol 2. Pol 3. Same, we'll look at the same kinds of things, what the promoter looks like and what are the basal factors. It turns out for Pol 3 we have 3 different categories or types of promoters, 2 of which, 1 and 2, involve internal promoter elements. This was the big surprise. For type 1 and 2, you can digest in the lab the entire promoter, what in most genes would be the promoter, all the way even into transcription. And transcription would always start a certain distance from an interior element back to here. Whether this was chewed away or not, it's always going to be reaching back to start transcription at that point. That's a favorite exam question. Experiment. I set up an experiment, I digest away, I see no difference in transcriptional activity. What kind of promoter do I have? A Pol 3. Although there is a class of Pol 3 that actually has a TATA and some more conventional upstream elements. Now you see these internal elements have names like box A, box B, box C. I'm not going to ask you to memorize the configurations here of the elements or just be able to distinguish between type 1 and 2. Just know there are internal elements in these first 2 types of Pol 3 promoters. Internal promoter. Deletion, you can still get initiation at a fixed difference. Deletion upstream and downstream to plus 55. That means you can chew into the transcript 55 bases and still it's going to start 55, a set distance from this box A. That took a while for people to believe. Oh, look, it's chewing, that's not me. What? I'm not doing it, people. And still no change in transcriptional activity. Of course, the other category, type 3, has some small elements that are actually upstream, like a more conventional organization that you see, and a site for OC, which is a transactivator, and PSE. Okay, commitment factor. Getting used to saying it. Commitment factor. Let's talk about it. Get this conversation over so we can go to the movie. Start point. Okay. What do we have here? We have these internal binding sites, and we have proteins that are called transcription factor 3A, transcription factor 3C, TF3C, TF3A. Unfortunately, and TF3B, not to be confused with TF2B. Notice the naming, transcription, these are how basal elements are named. These names don't necessarily correspond to the boxes that they bind, unfortunately. And because I can't remember it, I don't need to have you remember it also. Thank goodness I'm so forgetful, right? Okay, one of these, TF3B, has TBP. That is your clue that that's going to be the commitment factor. The type A and C bind to these internal boxes, box A, box C. Notice where TF3B is binding. It's actually binding upstream from the start of transcription. It turns out you first bind TF3A and TF3C, and they in turn position very precisely where the commitment factor should bind. And then the commitment factor determines very precisely where the RNA polymerase should start transcription. It's like these other elements are called, I think it's an assembly, let's see what they're called. Type 2, of course, doesn't have a box B. If you notice type 1, it does have a box A, a box C. This has 2 box C's. I'm not going to ask you to remember all those boxes. Assembly factors, yeah, I remembered correctly. A and C, those basal transcription factors that bind internally are called assembly factors, not commitment factors. TF3B is the only true initiation or commitment factor. C, you need to know some of the properties of these 3 basal transcription factors. C, it's huge. It's 500 kilodaltons, about the same size as RNA polymerase. A contains a zinc finger DNA binding domain. Of course, you don't know where the zinc finger is at the moment, but it's a eukaryotic DNA binding motif, which we'll study in greater detail a little bit later. A zinc finger incorporates a zinc ion to coordinate the position of its helices and beta sheets. It's not a helix-turn helix, but a zinc finger. It contains the zinc finger. What does B have in it? B itself, in addition to having TBP, it has these 2 other ones, and you'll need to know these also, because these are pretty cool. This one is easy to remember. BRF. It's transcription factor B, related factor. So even Paul 3 has something that looks like TF2B. It's B-related factor. It's a part of this commitment factor. It's there with TBP. Archaea has TBP, and right next to it there's a site for binding transcription factor B. In Archaea they don't call it TF2B because there's only one polymerase, right? So it's just transcription factor B, TFB. But they are often found together. Here they're in the same factor. So BRF is there in TF3B, which is the important one. And also B double prime, it seems to assist in melting the promoters. It's like a sigma function. So you don't have a true designated basal transcription factor that does melting like you do with Pol II, but that melting seems to be facilitated by B double prime. And I remember this by seeing that quotation mark. It looks like, to me, 2 strands of DNA. That reminds me that's the melting protein. So let's look at how things work. And I won't ask you to memorize this, but you'll see that this is a type 1 promoter. You've got 2 internal elements here, box A and box C. But let's back up a minute. This is pretty remarkable that you'd have an internal element, because this means, can this gene, is it free to mutate? What does it code for? It codes for tRNA. The DNA that encodes tRNA actually contains within it the binding sites for its own transcriptional regulation. If you mutate a tRNA, that's pretty bedrock. That's one of the foundation molecules of life. But here's another reason you don't want to, because it's also the binding site for its transcriptional regulation. The DNA is serving simultaneously 2 roles. Highly conserved, it's not mutating at all. This is what allows you to have an internal promoter. You could only have it in something that's highly conserved. And here you see it illustrated. So let's see what happens here. Well, A looks like it binds first to box A. It's easy to remember, but I don't ask you to remember. And C binds to box C. So far, everything is cool. And then B is positioned by the pre-binding of these 2 guys. They leave just enough room for B to bind. These are probably folded around, and it's attracted to these, and it must sit right next to them, although it seems like here is a big space, but these 2 position the commitment factor B. How about the type 2 promoter? Oh, there's no box C there, there's a box B. It looks like C goes down to box B, getting a little confusing, and C goes down to box A. Who's in charge here? And then once you get 2 copies of C, they direct the position, again, of TF3B. I will not ask you to be able to distinguish 1 versus 2. I just thought you might want to know how things work. What are the assembly factors? Assembly factor versus basal factor for Pol III. Let's see if I can pull them out here. Well, it's going to have 3. They all have TF3, so that's no help. It can't be B, because that's a commitment factor, so it leaves out choice 2. Can't be D, because we haven't talked about a D. That's in Pol II. It's got to be 3. Whew, man, we can continue. This is so stressful. Which of the subunits of TF3B assists in melting the promoter? Well, I'm looking for railroad tracks. I'm looking for double strands. I'm looking for that double quotation mark. B double prime. That assists in melting. And of course, finally, and this is where the trumpets begin and the heralds come out and they play those long, skinny trumpets with the banners. Pol II, in Her Majesty, has arrived at court. And this is where we began last time, looking at the structure of the Pol II promoter. One other thing we didn't mention was a silencer is the name of the DNA element that binds a repressor. What kinds of things bind in the enhancer region? Well, the transactivators bind, but they in turn, some of these transactivators don't have any ability to do anything except recruit even more proteins. Those additional proteins that are recruited usually have all the action subunits, the chromatin modification ability. And so transactivators usually recruit HAT activity, histone acetyltransferase activity, and complexes that contain HAT are called co-activators. They're called a co-activator instead of just a regular transactivator because they don't have the ability on their own to recognize a promoter or an element or enhancer. So you get the transactivators to recognize the promoter element, and they in turn now pull the co-activators which contain the action subunits. HAT in this case would be one of the things they do. What do the basal transcription factors do? Well, generally they end up recruiting RNA Pol II eventually. So that's kind of the simplified view of what happens. And of course, the same thing with repressors. Repressors per se are the proteins that actually recognize the DNA element, the silencer, and then the complexes that contain the action subunits, the HDACs, histone deacetylases, are called co-repressors. The same kind of logic. Steps in gene activation. Pol II, this is what happens in your cat. Step 1, transactivators bind. What are they binding? What is the state of the DNA they're binding? Is it all wrapped up in nucleosomes, or is it spread out beads on a string? There are several categories of transactivators, some of which have been called primary factors, or not primary, pioneering factors. These pioneer factors have the unique ability to recognize a promoter sequence when it's wrapped around the outside of a nucleosome. I mean, somebody has to get the ball rolling. These pioneer transactivators go ahead and scan DNA while it's still wrapped around nucleosomes, and they bind, and they in turn recruit chromatin remodeling and decondensation complexes to open it up so more generally transactivators can have access. So, the pioneer factors bind, they in turn attract co-activators, which have the HAT. Step 2, the transactivators open the chromatin up, and they do that not themselves, but because they have recruited co-activators that have the HAT activity. Step 3, these are busy little fellows, these transactivators. After they've opened the chromatin and recruited co-activators, they begin to recruit the basal transcription factors themselves. And what do the basal transcription factors do? They eventually end up recruiting Her Majesty RNA Pol II. So a lot of prep work has to take place before a eukaryotic gene can be expressed. People talk about two general types of transcription. Pretty soon we won't be talking about definitions anymore, but here's probably the last of the definitions. There's a couple more. It's the concept of activated transcription. Activated transcription is dependent on the binding of transactivators to upstream or distal promoter elements. If you just have the core, the basal transcription factors bound at the core promoter, you would definitely get transcription. But it would be called basal or baseline, basal transcription versus activated transcription. So this is to be distinguished from inducible transcription. This is not inducible. This is simply, it has the contribution of upstream transactivators. Or it doesn't have to be upstream. It could be from an enhancer that's inside the first intron, for example. That would be activated transcription. Any transcriptional activity due to elements and complexes that bind outside of the core promoter is called activated transcription. So, now we're almost ready to talk about TF2D, which we've already talked about. We're going to talk about the commitment factor and the other factors related to PAL2. Of course, they're called TF2 something, a letter of the alphabet. Well, I guess we're not ready to go there yet, are we? We're still talking about the details of the promoter. So you have the proteins that bind to the core promoter. Now let's talk about what these elements look like that comprise the core promoter. You know, in bacteria, you need to have a minimum of 2 elements to determine the polarity of transcription. Point A and B, so you can connect a line and determine direction of transcription. The same is true in eukaryotes. And often you'll have 2 elements. But sometimes you don't have 2 elements, and transcription will go both ways simultaneously. It is really a mess lots of times in eukaryotes. So one day anything works. So one of the elements that you have is it actually overlaps the start of transcription. It's not at -10 or -35. It's right there on both sides of point nucleotide 1. It's called the initiator region. It's right, and this is the consensus for it. C and an A, a lot of times it'll start on the A, but a pyrimidine, 2 of these, pyrimidine, 5 of these, so it's not a very tight consensus, but it is definitely recognized by some of the proteins of the pre-initiation complex. This is a general picture that helps you remember it. So here you see the start point, and you see this initiator region, CA, and it usually starts over the A, and these are pyrimidines on either side. And notice about 20, it's actually 20 to 30 upstream, so -20 to -30. Location is absolutely critical because you can find this sequence all over the genome, TATAA, as you can imagine. You can even find it in your promoter. When I'm asking you to identify TATA, that's a specific core promoter element. It must be located in that little narrow window, or you have to go, and it looks like a TATA, it's located at the right place, I think maybe it is. If it's located some crazy place or on the wrong strand, it's not a functional TATA in terms of promoter context. So you have the initiator, and then in some species, not so much in plants or fungi, but in Drosophila and some human genes, you have a downstream promoter element. This is sort of reminiscent of the internal promoters of PAL3, isn't it? But this is not in a highly conserved area that's actually codes for a protein or anything. A downstream promoter element, 20 to 35 or so downstream, a DPE. In addition, you can have our old friend, the TATA box, where TPP binds. Usually you don't have all 3 elements there. So you'll have initiator and DPE, or you'll have TATA box and initiator, but you always usually have some kind of loose consensus around the actual start point of transcription. In these that have initiator in DPE, they're called TATA-less promoters. 90% of promoters in the eukaryote have no TATA. That's pretty sad, isn't it? Yet you'll learn in various courses that this defines the eukaryotic promoter, and you'll talk about them as if they were the exception, the TATA-less promoter. No, that's the rule. Pol-2 promoters usually don't have, 10% have them. Is there a special category of Pol-2 promoter that is blessed with TATA? Yeah. You usually find it associated with genes that can be transcriptionally induced. So you wouldn't find it in housekeeping genes, but you'd find it in the heat shock genes for sure. You'd find it in those that are induced in response to pathogen attack or developmental triggers, developmental cascade of embryogenesis. A lot of these have the TATA itself. Most genes do not. In fact, some genes, it's hard to even see an initiator. It's hard to see where the promoter is, but we know it's the promoter because transcription starts there all the time. Some of the promoter seems to be controlled perhaps epigenetically by modifications of the histones. We're just still unraveling what we thought was simple. It's turning out to perhaps be a little more confusing. Minimal promoter. It always includes the initiator, always in quotation marks, in either a TATA or a downstream promoter element. Anyway, it's those sequences very close to the start. How about commitment factors? Well, this is kind of a review. You see here all three promoters. This kind of sums up the commitment factor idea. So let's see what the slide has to say. For Pol-3, it's TF3B. You remember the different subunits of 3B. BRF looks like B, B double prime melts, and TBP itself. For Pol-1, the commitment factor is SL1, contains a couple other proteins, and TBP. Pol-3, it is, we talked about that. Pol-2, it's TF2D. It's the one that contains TBP plus 11 to 14 associated subunits called TBP associated factors or TAFs. Oh, these are the last of the definitions right here. Basal factors, we've talked about. Upstream factors, anything's not a basal factor, right? Inducible factor, an inducible factor, that doesn't mean that, it's not a factor that mediates gene induction like you'd think. It's a factor that starts out, a positive transcription factor. It starts out without any ability to act. It's like an APO or something. So the activity of the transcription factor has to be activated or induced itself. The heat shock transcription factor is an example. It doesn't bind to its elements and actually activate heat shock genes until it is activated itself. The estrogen receptors are inducible factors. They have to be rendered, they go from an inactive state, and when they bind the steroid, now they become active. That's called an inducible factor. So when the induction doesn't, we're not talking about the transcription of the gene they regulate, but their ability to act in a positive way, inducible factors, and that's the one that's not quite intuitive. Okay, minimal promoter, you've already got all that. Enhancers, they can have a scattered distribution. Here you see these upstream elements here. They can be all scattered or they can be clustered together in what's called an enhancer. Enhancers, one of the terms is module. An enhancer will have modules, which are really just upstream binding sites for these transactivators. activated transcription involves the participation of these distal promoter components, whether they are distal upstream or distal from the core in a downstream configuration. If they are participating in the activity you see, it is called activated transcription. Here you see, we've talked about the transactivators, whether they're in an enhancer or in isolated elements, recruiting chromatin modification complexes, the hats. There's some others they recruit also, but here's just kind of a crude illustration of them recruiting basal transcription factors. And the basal transcription factors in turn recruit RNA polymerase. Many, if not most, of the basal transcription factors remain at the promoter after the RNA polymerase has left. You know, it took so long to get them all there. Why break up the band? Most of them stay, so the next round of initiation can go much smoothly, or quickly. That's the end of that first part of transcription, introduction to the big picture. What's the time? Who knows? When you're having fun. I'm going to close this down and go to part 2.
An hour.
Well, hour? Oh man, can anybody stand it? Well, you're still here, I guess. We'll get through this together. Oh, I'm going to talk about TBP. I choose some of the weirdest things to talk about. What, is this whole thing going to be TBP? Well, let's see. Universal factor, and we've talked about it being in each of the commitment complexes here. Here we're beginning to talk about specifically down here, TF2D. 2D contains TBP, and it has 11 to 14 associated factors, TBP associated factors called TAFs. Many of you have TAFs as your unknown for one of your gene bioinformatics projects. It forms a saddle around the DNA and bends it by 80 degrees. What else bends DNA when it binds? Cap. We don't know why that has any influence. We don't know why this has any influence, but it certainly does bend the DNA. And this is what it looks like. This is a wireframe configuration of the DNA, and you're looking head on. There's a drill. I don't know whether probably none of the questions are hooked up to the homework quizzes. When this is the first time the course has gone up with the UF Online, so I just haven't had time. I'll need to go back today or tomorrow morning. But I have a drill there that gives you different orientations of TBP, this is TBP, and DNA, and I want you to look at it because I'm going to ask you, I promise, on the exam, you're going to tell me the direction of transcription. Whoa. Yeah. And I'm going to show you how to do that in a minute. You're going to look at this. But look at just TBP, this molecule we inherited from archaea. Notice its symmetry. Beautiful symmetry. These are called stirrups. This is where your feet go. And this is where you sit. Be careful, that N-terminus can be troublesome. But this is a saddle, maybe that's the horn of the saddle. And notice if you split this down the middle, that both ends, the C-terminus and the N-terminus, are on the same side. It's like somebody with a hair lick in the hair. You see the N-terminus, most of the N-terminus is over here, but the end of it goes over like that. It's on. So you see the side of the molecule with the two ends, the two strings. That tells you the C-terminal half of the molecule versus the N-terminal half of the molecule. Why would you ever want to know that? Because the next transcription factor, which is 2B, binds only right down here on the stirrup of the C terminus of TBP. And that's important when you're trying to figure out which direction transcription is going. So if you think of this, well, we'll talk about that later, but you see the hairpins up here, and then somehow the different tafts find a place that they can grab hold. This goes all the way back to Archaea. And this is the third example and last example of binding to minor groove. Tata binding protein. It doesn't even use a helix. You see this? You can't see it on this view, but this is a beta sheet. If you turn this up, you'd see this is a beta sheet that's sitting in the minor groove. Very unusual. Highly conserved. We still have it. N-terminal stirrup is where TF2A binds, C-terminal stirrup, TF2B. Think of it as a saddle. Place to sit. This will help you determine the direction of transcription. Because if you can sit in it as a saddle, then you are looking in the direction of transcription. All you need to know is where you put your haunches, which is right up here on the springs, and where to put your left foot. You always put your left foot on the C-terminal stirrup. So in this picture, you're sitting on it like this, and transcription is going towards you, out of the screen towards you. Learn to ride. So I don't show people how to ride, as freshmen and sophomores, because you can hurt yourself trying to get on this thing. And when I gave paper exams, there would be people in all sorts of contorted positions in the chairs as they're trying to stick their feet into the saddles. A lot of injuries resulted from those who hadn't been practicing at home. Now we have Proctor U. Those proctors were looking with them with puzzlement. Going, I can't see your eyes, I'm seeing something else. I'm going to give you this in all sorts of crazy orientations for you. And you're going to have to do the mental gymnastics. All you need to do is where to sit down and where to stick your left foot. Once you're in the saddle, you can tell me which way transcription's going. Okay, so a little more to get acquainted with the adversary here, so you can recognize this thing in the dark, which is, of course, how the nucleus has to do it. There's not a lot of light on your spleen at the moment. Everything's working perfectly fine, I think. This is what it looks like looking down on it. This is DNA underneath. Looking down on it, you see this beta sheet? It's sitting kind of slantwise in the minor groove. The N terminus is here. The C terminus is there. How can I tell? Because that's where that end is, and there's another end. That's where my left foot goes. So if I sit down here, transcription's going that way. Let's see here. This is showing the bending here that happens when the two phenylalanines insert. It's really quite a bend. What else do I have here? Okay. This is looking up underneath like you're changing the oil on your pickup truck. You're underneath and you're seeing through the DNA, that's this ribbon, and you're seeing that beta sheet from the underside. That's the underbelly of TBP. Which way would transcription be going there? Oh dear, well, let's see here. That looks like an end. This must be the C-terminal stirrup right here. So if that's the C-terminal stirrup, but I'm sitting in the back like this. No, I'm sitting like this, because I got to get my left foot in the C-terminal stirrup, but I got to get my haunches on the other side of the screen. So I'd be sitting like that, right? This is not on GoPro, is it? So that means transcription will be going that way. Here you see it sitting in the minor groove. View from upstream looking back. You can see this is the easy one. If you sit here, your left foot's here, you're just riding like a normal day on the ranch. Okay, TBP. There's a drill there that puts in all sorts of orientations. You can open up that and do it as many times as you want. You can't get but like 1 point or something from it, but I would recommend that you do it because you will be asked on the exam to tell me the direction of transcription. But there's another clue I'm going to give you. If you find it hard to identify the C-terminal domain, the C-terminal stirrup, transcription factor 2B knows where the C-terminal stirrup is. So if I give you TBP and TF2B in a complex, you're always going to put your foot where TF2B has bound. Easy peasy. So here you see TF2B as a segue. TBP on the left side. This is TBP's in yellow. It has, this would be the N-terminus. It has what used to be drawn like this, and they called it a B finger. And this B finger, this actually inserts into the interior of RNA polymerase. This is that magical 3.2 domain that interacts with the 5' terminus of the nascent transcript. This was called the B finger. Now we know much more detail its structure, and the B finger is not appropriate anymore. But it has a zinc protein-protein binding domain they call the zinc ribbon. But notice right here it's got repeat 1, repeat 2 at the C-terminus. This is highly conserved, and this looks like evolutionarily, or maybe it's the other way around. It looks like a cyclin, the subunit that regulates kinases. 2 cyclins have joined together to make this TF2B, but TF2B goes all the way back to archaea, so maybe TF2B came first and cyclase came second. The zinc ribbon here is what is a dock site for RNA Paul II. Her Majesty, the Queen, Paul II, binds at that site, among several things. That's a mess, isn't it? You're going to see this kind of detail on exam 4. You love this, right? Easy peasy, pictures. I'm going to have arrows. You're going to tell me what you're looking at. It's like, okay. This is the N-terminus. There's that zinc ribbon. And what you see here is a B reader helix. And then you see a B linker helix. No, linker, reader, ribbon. And of course, this is the beginning of one of those cyclin domains, repeat 1, repeat 2. And notice how this is now superimposed on the RNA polymerase itself. See how this thing snakes on down in here? This is what is snaking on down and making contact with the initiator element and with the 5' end. We'll see that in just a minute. Here you see TBP. Notice how the DNA is coming like from my direction, and then it makes a 80-degree turn. And so you see TBP, and notice on the back side here, you have the 2 globular domains of TF2B, right? and then the N-terminus that snakes down into the interior of RNA polymerase itself. So this is B. Direction of transcription is that way. If you were sitting on this, your left foot would go here, and it'd be like on a roller coaster, you'd be getting ready to go down that way. The start of transcription will be right there. That's why this, in its specific interaction with polymerase, determines where transcription must start. That's why it's so constrained, because this fits together in a big jigsaw puzzle. And so here you see the melting beginning to occur, and the B linker participates in the melt. This is a sigma-like function. The B linker. See it up there, getting ready to melt? It doesn't melt at exactly +1. It melts at about, what, -10 or so? The B linker, the B reader, the zinc ribbon. The zinc ribbon's attached to the wall of the polymerase. The B reader is going to be interacting with the nascent transcript as it grows and be responsible for that abortive cycling. Here you see even more deep. This might even be on exam. I don't know. I'm so forgetful. B linker. This is melting. B reader. Notice how it is this is the RNA, this is the active site, this is the 3' end where it's growing. This is the 5' end of the RNA here. The B reader is interacting with the nascent, or in process of formation, the nascent transcript at its 5' end, just like sigma domain 3.2, and that is what gives us abortive cycling, even in a eukaryote. B reader contacts not only the nascent, the 5' end of the growing RNA, but it contacts in the initiator at -8, before there's any transcript being formed. You see more detail. The rudder, rudder is what splits the RNA from its template. And this is a comparison between TF2B and sigma. You see no similarity when you look at the actual amino acid sequence. But when you look at the structure, you see a resemblance. (3.2). Okay. You see this is highly conserved in lots of things. Here's some more. Apparently, I just love this. This is the B reader right there. That's the loop coming off the end of it. And you notice that B reader, it's interacting with the 5' end of the transcript, but it's also bending it towards the exit tunnel. That poor baby transcript is coming up. It's just been born. It doesn't know how to get to the TV room. B takes it on by the head, pushy back, okay, let's go. It leads it to the exit pore. Diverts it, just acts as sort of a guardrail. And that's as much detail as you'll need to know, thank goodness, of TF2B. But you will see some of those structural diagrams on the exam 4, and I'll have arrows pointing. The kinds of things I'm going to ask you to identify are the B linker, the B reader, the zinc ribbon. But I might not say where's the link, I'll say which domain is involved in melting. I could ask you, well, you have to know something before that. Now this is back to helping you with the question on how to ride this thing back to the rodeo. Because this is actually what it's going to look like on the exam. It's going to have TF2B there also. And TF2B is always attached to the C terminal stirrup. So if you just look for the green blob, except it may not be green, but it'll be a blob, versus this highly symmetrical tata binding protein, right? That is where your left foot goes, and that's where your haunches go. So the only way you can sit on that is facing you guys, coming out of the screen. What does TF2B do? This is sort of summary. Linker melts, the reader recognizes -8 of the initiator and contacts the B loop, thus it comes out the end of the reader, contacts position -1. It also is what interacts with the 5' end of the nascent transcripts and initially blocks its exit. Okay, so we're moving now away from B. We've talked about TBP. We've talked about B binding to the C terminal stirrup. How about the right foot on the N terminal stirrup of TBP? That's transcription factor 2A that binds there. So you have, I think of these things as saddlebags. You got your saddlebags over here. On the left, your C terminal stirrup. And this is where you put like a knight, a medieval cowboy, I guess. You have this lance. The lance is that probing N-terminus of B that protrudes way down into the active site of the polymerase itself. That's on your left. On A, you've got these two saddlebags, TF2A. So you end up with complexes on both stirrups. And you end up kind of engulfing the DNA on both sides, and it stabilizes the interaction of TBP with the DNA. At which position is TF2B attached? Uh-oh. Where is B attached? To the C terminal stirrup. Which of these would be the C terminal stirrup? You're looking for where the ends are. That's the C terminus, that's the N terminus. This half of the molecule is the C terminus, that's the C terminal stirrup. That must be position 3 is where TF2B would be. Just love TF2B and TBP apparently. More slides. More opportunities for me to say, what is this arrow pointing to? So this is RNA polymerase, and you're looking down sort of from the top. That's TBP. Okay, you're sitting like this, and on your left foot, this yellow is TF2B. These are the two globular cyclin-like domains, and notice the N-terminus snakes down underneath the DNA into the dark interior of the active site. If you rotate this up, you can look down on TBP in green, and then on the left stirrup, you see the globular domains of B, and then you see the N-terminus snaking down underneath the DNA into the interior. TBP positions B. B positions Pol2. TBP and TF2D itself is called the commitment factor. Why don't you call TF2B the commitment factor? Because they don't. They call TBP because it's involved in all the others. But you know, PBP has something that looks like B also in the Pol3 promoter. So that these two guys kind of work together. Here you're looking down again. It's the C, the yellow is B, TBP. N-terminus. Sometimes on an exam I'll have an arrow pointing there, and it's the N-terminus of 2B if it's snaking down into the interior of the polymerase. This is the Mr. Potato Head model of Paul II. And so this is one where they've taken B and split it. It's still yellow, thank goodness. They've split it into the N-terminus and C-terminus. And so the C-terminus is here, and the N-terminus is there. When it's all put together, this is the N-terminus that's snaking around from the purple underneath the DNA into the active site. That is B. What is this? Oh, we have other things here, don't we? We have some blue, and we've got some orange, blue and orange. What an odd color combination. This is TF2F, which we haven't talked about yet. And this is TF2H. Remember H involved in nucleotide excision repair? See how an integral part of transcription it is, yet it's used in nucleotide excision repair? Could you identify, if I had it, sometimes I point at that thing, that yellow thing, and people go, what? You can't possibly think I know what that yellow thing is. But also F, it has domains labeled 2 and 3. People don't talk about that much anymore, but these have some vague resemblance to domains 2 and 3 of sigma, TF2F. I'm hoping I get rid of those kinds of questions. Oh man, this is like a snow cone or something. Updated model for the pre-initiation complex of RNA Pol II. This is where we're headed. You know, the holoenzyme in E. coli. This is everything there? Nah, it's got even more stuff. This is a simplified view of what happens at the Pol II promoter. More than 50 proteins come together. But so this is based on the EM topology technique that's growing in importance. Here you see the two shades of blue being the DNA, TBP, F, H, E, B, it's a met, and this RNA polymerase subunits 4 and 7. They play an important role with interaction with somebody who's not here. There's another transcription factor, and you're not going to need to memorize this at all. There's another transcription factor that binds here and covers this whole side, just dwarfs it, that's used at every promoter, called mediator. That's where you play the kettle drum. Mediator, boom, 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 boom. Mediator, first discovered in yeast. Okay, so this is as far as Lewin. This is what Lewin thinks. What's wrong with Lewin's perception? Lewin made a mistake here. I don't know whether this figure's still in your book or not. It might be. But notice he's got TF2D. Okay, kind of looks like a blob. Exit 4, got the DNA. He's got TF2B, N-terminus, C-terminus, enzyme. What is wrong? He's got the N's label wrong, doesn't he? That's the C-terminus. Everybody knows that but Lewin. So he's got those two labels mixed up. What else does he have wrong? He's got a gap in his knowledge. He's not going to be well prepared for exam 4. Notice the N-terminus doesn't snake down under the DNA. It sits on the top. That's not right. It curves down underneath the DNA. Doesn't lay across the top of it. Oh me. Poor Lewin. Now this is called a color clash. You have an intense red against an intense blue. It's highly disturbing to normal people. It looks fine for highly disturbed people. It's a thing of beauty. It's just there to disturb people. Okay. The mysteries slide. Look at that. TFLLB, that's like a shoe, isn't it? Or something. TF2B is not required for cell viability. What? Something that we inherited all the way back from archaea is not essential. Then why do we still have it? Let us read further. Less than 1% of the coding genes have an altered expression by twofold when you completely silence TF2B. Less than 1% of the protein encoding genes have any change in their expression if you remove something as essential to this lecture as TF2B. What the hey? Why did I put this here? Many viral promoters require and is involved in cellular differentiation. Differentiation is pretty important for metazoans. How does transcription function without a protein to melt and align the start site for transcription? I don't know. That's what you guys have to figure out in your generation. Are there other proteins in the cell that substitute? How does transcription work in your cat? In you? In your big toe? These are all questions. More mysteries. TF2B associates with tubulin and with mitotic chromosomes. We don't know why. It works in mysterious ways. B is present in the termination site of the transcript. Termination is involved in termination of transcription and is required for gene looping. That's where the 3' end of the gene circles around like a big horseshoe and joins the promoter. That seems to be the natural configuration for the gene as it begins to be transcribed. The end point is making contact with the start point, and TF2B is part of bringing together that happy circle. Phosphorylation of B is required before you can recruit the add poly-A complex. Here you see a model of the end of transcription being joined around B. It's a very crude model at the promoter. So there's still a lot to learn. Our lab was the first to clone TF2B in plants. Took the student 9 years, but we did it. And nobody cares. Okay. So let's see how this works here. The first thing to happen is TF2D will bind the promoter, binds in the minor groove, and then you have A, B, F, and E joining later. This is from the book. Paul 2 comes later. Of course, there'll be more details in a minute. But we have some more transcription factors to talk about. E and H. These are involved in melting. Wait, okay, they're involved in melting. Maybe that's how cells can melt if they don't have B. H got 2 perfectly good helicases. Okay, so you remember that C terminus CTD of Paul 2? That has to be phosphorylated in order to trigger the transition from initiation to elongation. That's one of my favorite questions, right? What's the trigger? In bacteria, it's the pushing of domain 3.2 of sigma out the RNA exit pore. The loosening of sigma allows the promoter to finally move on to nucleotide 10, 11, 12, 13 and start elongating. In eukaryotes, that's something even in addition to that needs to happen, and that is phosphorylation of the CTD. The phosphorylation of the CTD turns that CTD from just a random peptide into a binding site for the capping complex. Cap complex can't bind unless you phosphorylate the CTD. Of course, the capping complex binds the CTD. Phosphorylation of the CTD required to end abortive initiation. PTEFB is the kinase. How much time do we have?
You got 20 minutes.
20 minutes, okay. So I think we're going to go into the next series of slides. I haven't seen this in a while. Goes into the multifunctional role of the CTD of the largest subunit of Paul II. And so here we go. Phosphorylation, here you see the capping complex binds to the phosphorylated CTD. These little green marks represent the CTD and it's phosphorylated. Then you get scarfs. This is a intron processing, splicing complex, and then you have polyadenylation and cleavage complex that eventually joins. These are the major complexes that bind to the carboxy-terminal domain of the polymerase. Cap, intron splicing, termination poly A. The CTD has to be phosphorylated before these complexes have affinity. So I believe there are questions in homework, maybe on the exam, where I'll give you a list of things and say, which of the following is not a complex or a function of the CTD? I hate those kinds of questions. But nonetheless, they remain on my exam. I don't know how to get rid of them. I guess delete would do it. But so kind of get this in mind. The main ones are the first three here. This capping, the 7-methyl-G, the SCAF, which is splicing, spliceosome, polyadenylation, and cleavage of the transcript. That's how the transcript is terminated in a eukaryote. The polymerase doesn't do it. There is no intrinsic terminator. It's actually cut. And so that add poly A signal you see in the 3' untranslated portion of the messenger RNA is there for two reasons. To cause the complex to cut at that point while the polymerase continues on, and then at the cut site start adding several hundred A's. Which of the following is not a which of the following is not a function of the CTD? Melts the promoter. That's an easy one. Connection between transcription and repair, and we've talked about that. Remember we talked about the MFD in bacteria, whereas it's H that recognizes it, it recognizes a stalled polymerase. I think we've already covered this types of stuff. Demethylation of DNA at the 5' end is necessary for transcription. Okay, next topic here. I guess this is following the order of topics in the textbook chapter. So now we haven't really talked about some of the other basal transcription factors. Remember, we still have H and mediator to talk about. But I think the book takes an interlude here and talks more generally about promoters and the role of DNA methylation. If you methylate a promoter, you often inactivate that promoter because you make the promoter elements there unrecognizable by transactivators and repressors. So if you demethylase, You usually activate a gene. What is the site of methylation? Is it any old base? Well, there are lots of bases methylated, but predominantly it's C. But it's C in a particular configuration. CpG doublet. CpG doublet. What does why do they put the P there? That is to convey orientation. 5' to 3', CpG. So that's the top strand. What would this look like on the bottom strand? It would look like a G and a P and a C. But you wouldn't read it that way because the bottom strand's going the other direction. It would still be CpG on the bottom strand. So both strands are methylated at the C. There'd be a C here. And if you read it 5' to 3', it always says CpG. It's the C that's methylated. So if you see this doublet, a C and a G, and it has to be 5', 3' CG, that is a site, potential site for methylation. You will see clusters of these sequences. You begin to look at eukaryotic promoters, and the first thing you usually scan for is a tata. It's like finding a 4-leaf clover. Only 1 out of 10 is going to have one. You feel lucky I found the tata, but most of the time you're not going to find one. Or you'll find one at a crazy place and it says tata, but it's not functionally a tata. So what other easy peasy things can you glean out of the sequences? One is the clustering of CG when you're looking at the top strand, because the top strand by convention is the one that's reading 5' to 3'. If you see a CG, then you see another CG. Sometimes you see them clustered in the very rich regions called CpG islands. Other times you see them scattered through a promoter. A lot of times you can control the activity state of a promoter simply by putting that little doublet there. It's a crude control. Methylate the promoter, it can't work. It's not responsive on a short time frame. CpG islands surround all promoters of constitutively expressed genes where they are unmethylated. See, this is the non-intuitive part. This is exactly what should be methylated, right? CpG. You see CpG islands all around promoters, but you don't see them methylated. They are deliberately protected from methylation. That's their control function. When you stop protecting them, they become methylated and they are silenced. So it's kind of counterintuitive. You see the CpG islands, you go, this must be the place that's methylated. Yeah, if it could be methylated, but it's usually clustered around a promoter as a control device, a very crude control method in eukaryotes. Many, many of our genes have this crude level of control. You see how widespread they are. There are 29,000 CpG islands in the human genome. Of course, some of these mites, transposon leftover, the ALU repeats have CpG islands also. Of course, they're shut down all the time and they're heavily methylated. 10,000 conserved between mice and men. Are they regulatory? We're just beginning to appreciate their value and their role in regulation. Repression is caused by proteins that bind to methylated CpG doublet. So the methylation of the C in the CpG doublet acts at 2 levels. One, if this is in the middle of a transactivator binding site, it usually prevents the transactivator from binding there. On top of that, there are control proteins which specifically bind to a methylated CpG doublet. And those proteins attract complexes that shut down the gene. So you prevent activation, and then you recruit things that actively try to repress. And these are the proteins, the key words, that bind to the methylated CpGs. Methylated C protein 1 and protein 2. MECP1 and MECP2, they bind, and these two in turn attract complexes that condense the chromatin and repress. What does it bind? Okay, so we've talked about HAT or HJAK. We talked about HAT complexes, and generally we call them co-activators. Then we talked about repression. You'll have a repressor that binds to a silencer, and it in turn will recruit a co-repressor which has an HDAC. SYN3 is the name of a specific and widespread and conserved HDAC co-repressor complex. SYN3 is one of the subunits of a larger complex, but it contains HDAC. It has affinity for MECP2. So this is an example where these proteins which bind to the methylated CpG doublet recruit a specific complex that contains an HDAC. So in a sense, they would be repressors in a functional sense, MECP1 and 2, because they recognize the DNA directly and they recruit an action complex. And this would be a co-repressor. SYN3 would be the co-repressor component. Of course, that's what HDAC does. It removes acetyl groups, and that ends up a series of as yet fully understood steps condense the chromatin, repressors. Here you see the, let's see what the clusters. I think what you're looking at are the doublets represent the lines, and if the lines are closely spaced, they represent clusters. So you can see for an island, by definition, it has to be at least 10% of the bases have to be CG, not GC, but CG. That is enough to comprise an island. Notice how long they can be, several thousand bases in length. The exceptions, Drosophila, other dipterans, which are flies, have very little methylation. How can you make a CpG island work if you've got no methylation occurring? Nematodes, there is no methylation of their DNA. We don't know. Methylation CpG island seems to regulate which types of genes. It was kind of a crude level control. It's constitutive genes. What is constitutive? Constitutive has the same sort of unregulated activity. Chugs along, housekeeping genes. They're not regulated up and down. But this is a way to regulate a constitutively expressed gene, which is most of the genes. I hope that's the right answer. I've got so much invested in it here. Let's see. Whew. Which of the following proteins binds to it? This is so easy. MECP1, MECP2. The end of part 2, I think that's a good place to hang up the hat today. How much time we have? 15 minutes?
15 minutes.
About 15 minutes? Can anybody stand to sit here? The haunches cry out for exercise. Let me walk. Let us walk. And we'll get into the other. There's too much. The head's exploding here. We'll take up the Part 3, next time, eukaryotic transcription. There's a lot of material here. I think that's about all people can absorb at one sitting. Maybe too much.